you are make the surrogacy, then supervision of the donor, synchronization of recipients, flushing of the donor, searching and grading of embryo, transfer of embryo, freezing of embryo, thereafter donor and recipient management. So here you have to take the cyclic donor, that is the important for better result. And the 9 to 12 day mid cycle is the best days to start this protocol of hormonal treatment. This slide are not moving again.
Sir, I think you can just try moving slide.
Not moving.
You can reshare it, and after your session, we will share your PPT with the participant. You just kindly share PPT with Dr. Dinesh, and we will share the PPT with the participants also.
Okay, okay, ma'am, that's fine. So I just 1 minute.
As of now, you can reshare.
Yeah, stop share and I will try once more.
Yes, sir. We are still on the same slide.
Uh,
Major steps in MOET.
just 1 minute, ma'am.
Sir, have you shared your PPT with our team?
Not it, ma'am. Just 1 minute. Uh, I think they have not requested also to share. Now it is moving.
Yeah, now it's moving.
Uh, mid cycle is the best time to start your superability treatment. And this is the skeleton which we are following at our farm for producing that in vivo embryo, where generally for Sahiwal cow we found that day 9th is the suitable day, where morning and evening we are giving the hormones. Then 10th day morning and evening or noon time we are giving synchronization with 1 dose PG to recipient also. Then, along with fifth dose of FSH, we are giving PGF to alpha. Then day 11th, we are giving FSH. Day 12th, also morning and evening. Then animal invariably donors will be heat on day 13th, or we can call it day 0. And thereafter, we are on 7th day, we are going for the flushing and then PG. And this is the dose we are using. Our boss indicus animal are very much susceptible to the gonadotropin. So they are 150 to 250 micrograms are enough. Earlier stimulol were available. Now it is not available in India. Faltropin being used extensively, which now we are also using this one only faltropin, where dose is more or less similar to the similar, but here the availability is in milligram and their availability is the microgram. Then our crossbred animal require little more hormones. Then our purebred animal also require more than our Indian crossbred. And our buffalo is taking more one almost 1 vial for give proper stimulation. So in market currently only two products are available. Two products are available. One is the Foltropin B, another is the Stimifol, especially in India. So these are these two products which are being used, and these are the things we are doing at our farm. And farmoid, these are the materials being used. Like you require catheter, you require flushing tube, media, and filter, then pipette, stereosome microscope. And we are doing also under field condition. It's a very easy techniques you can perform under field condition. Also, only you have to have one stereosome microscope and some plates, heating plates for making the isothermal environment for embryo. And definitely, when you search the embryo, you have to grade which one is the grade 1 embryo, grade 2 embryo, and grade 3 embryo according to the IETS guidelines. If there is symmetry of zona pellucida, and spherical embryo mass with individual blastomeres uniform in size, in color, and density, Irregularity should be minor. Mm, Cellular material at least intact, 85% viable embryo mass, zona pellucida is smooth, no concave. Fair moderate irregularity and overall shape of the embryo for grade 2, and this grade 3 embryo are poor major irregularity in overall shape and embryo mass, color density, and blastomeres. Um, so up to grade 1, 2, and 3, you can go for fresh transfer. And for freezing, 1 and 2 is the good. Better not to transfer this grade 4 embryo. So, seeing these hormonal protocols, it require a lot of animal handling and manpower. So, scientists started working to make some sustained release FSH. So, in that context, this Japanese scientists, they have made one entrin R10, wherein I think they are making aluminum hydroxide gel for slow release of this one, and they found that it is giving satisfactory single dose injection itself giving satisfactory result, and they found that entrin and our conventional FSH formulation, more or less number of transferable embryo, there is no significant difference. But still, people are preferring this our conventional methods, those who are practicing in vivo embryo production under their farm condition. So, really, this regime, single dose regime is the need of hour. So some scientists they have tried with poltropin B and hyaluron at this concentration of 20 milligram per ml would appears to be effective. Superestimation in our Angus donor cow with a single intramuscular injection of poltropin B diluted with 20 milligram per ml of hyaluron resulted in a comparable superveterinary response to the traditional twice daily intramuscular injection treatment. And reduce definitely if you are going for the single injection with hyaluron or aluminum hydroxide gel, so definitely your labor handling and animal welfare will be protected. Though, so during summer season, during summer and especially rainy season, we found that there is a compromised embryo production. So we tried with additional supplementation of flaxseed at the dose rate of 300 gram flaxseed per 100 kg of body weight, and we found that these flaxseed supplementation have improved the ovulation rate during even off-season breeding, and also there is a less anovulatory number of anovulatory follicles, and other parameter are comparable, and also so you can see here animal with good supervilatory response, very less number of the anovulatory follicles, and here animal with poor supervilatory, most of the poor supervilatory response. So through ultrasonography what you found that majority of the your dominant pre-ovulatory follicles get luteinized. So after supplementation of this omega 3 source, especially our flaxseed, so the chances of anovulatory reduced. And if you see our result, see the result, if you show excellent and good quality embryo are, are more in supplemented group and hydrated group, so significant difference per flushing, and number of dead and degenerated are very less. If you that you so supplemented animals having more good quality of embryo in picture you can visualize, and here if you see that inner cell mass is little bit scattered, loosely associated. So and also definitely, and when we tried with conception rate, however, there is no significant difference because our data is in small animals. Though numerically there is a 10% higher pregnancy rate in a treatment group. Then again, challenge is there how to select the donor for our MOIT program or MOIT supervisory program or OPU IVF program. So based on the literature, and now we are also practicing, we are seeing that blood flow to the ovarian tissue. That too specific to the medium-sized follicles. If there is more blood supply, more number of follicles are tinkling with some colors, definitely they are estrogenically active and going to either good oocytes are going to produce good embryo if in practicing in vivo. So in that context, we started and we found that ovary with good blood flow are giving better result as compared, and we found that there is a good correlation with if more number of large follicle on yes, so it definitely will get more number of CL and more color follicles are there, definitely we're going to get more CL and more color follicle are related with more number of embryo and uncolored related with this one. And strategy for donor selection. So we thought that what could be the criteria. So we tested and we started 20 and above for our indicus, especially Sahiwal and Thar Parker cattle is suitable with good production history production performance suitable to take our MOIT programs. And we found that in high FC animals, they are giving more number of embryo as well as their quality is also very good as compared to the control as compared to the animal with low FC, enteral follicle count. And what we found that dead and degenerated embryo also very less in this animal with high AFC. And we found that this very positive correlation of enteral follicle count with our number of embryo and number of CL, corpus luteum. So here you just imagine a picture from our farm only. So so see here, I think 14 embryos are there, 14 to 15 embryos. Most of the embryo are grade 1. And animals, donor from a low FC group, we found that very few embryo are good quality. So high FC, that should not be too high also. Sometimes there will be overcrowding and may lead to the nutrient deficiency and poor development of the embryo and quality will be compromised. So there should be a balance between the AFC count. So you should select between 20, 25, 35. I think after 40, 45, there will be crowding and problems with embryo production. So that finding we have published in our one frontier, frontier in veterinary science. So enteral follicle count affect the luteal characteristics, superovulatory response, and embryo quality and Sahiwal cattle. Then in both Taurus animal and the purebred animal, there is a problem of dominant follicle. So we tested in our indigenous cattle, though data is small number, about 14 animal in treatment group and about 12 in a control group. So we found that this dominant follicle doesn't affect the embryo production in our indigenous animals. So thereafter, we are not going for either aspiration or ablation of the dominant follicle. If it is a present, also we are proceeding either for pickup or for flushing. But there is a one crucial issue. If your progesterone concentration is more than 1 nanogram per ml on the day of superovulatory stress, definitely that animal is going to yield a poor embryo, and also quality of embryo will be very less. Number also will get compromised. You will get more number of anovulatory follicles. So DF presence does not impact much in our indigenous cattle. However, elevated level of progesterone at the time of superovulatory stress are hampering the quality embryo production prospects. Then we also correlated the AMH concentration. So AMH is very much positively correlated with animal with high AFC. So this is the photographs when you are started your protocols. Then a small follicle being converted into the medium-sized follicle. On fourth day, medium-sized follicle converted into the large size of the follicles, pre-ovulatory size of the follicle. Then on the day of flushing, you will get more number of the corpus luteum in the either or both ovary. Definitely in each flushing or each superstimulatory protocol, some few anovulatory follicles will be there. So whatsoever you are getting through ultrasonography, it is not perfect. Means because ovulation will happens all across the ovarian surface. It's not a mere where ovulatory foci is the point of ovulation. So through ultrasonography you are getting only two-dimensional image, and through so for counting your superovulatory response, you have to palpate the ovary all across the surfaces of the ovary, not just seeing the ultrasonography and telling, okay, it's a poor response, it is good response, or it is moderate response. So here few animals are there where we prepared everything, but we didn't get any embryo because of too much number of anovulatory follicles. So one day before your preparation or taking animal to the sponging chute, you have to confirm whether sufficient number of corpus luteum are there or not. Then only you go for flushing. If sufficient number of corpus luteum are not there, better to drop your flushing program and give only treatment to the donors for PGF12 and other treatment recommended for next cyclicity. So what we found that after giving the PGF12, within become normal for the donor. And day 5, ovarian size is also reduced, almost normal in size. However, if you see because of multiple ovulation and more number of superovulatory follicles, size is become almost double or more than double. Usually, its size vary between 19 to 22 But when you are going for the superovulation and all, it's become sometime 35, 34, even 41, 42 also. So just double, but within 5 to 6 days, almost recover to the normal ovarian size. So here just luteal blood flow. Actually, so we tested after flushing most of the animal, 99% animal, they are coming in estrus in 1 week. So we doesn't go for AI in those estrus. So we wait one more cycle and we go for the insemination. So nothing is much harm. So from few cycle we have recovered extra more number of the embryo, 17, 13. So that's because our indigenous animal are very much susceptible to the gonadotropin treatments. That kind of figure from the buffalo, our crossbred up to red. So seeing that hormonal protocol, time taking, and where you have to wait the animal in the mid cycle, either you have to create the artificial corpus luteum. So there are certain limitation or critical factors before starting the in vivo embryo production. So people switched over to the in vitro embryo production, and it is more intensive. However, it requires much more technical manpower, a good laboratory facility, as well as hygienic environment and efficient technician to handle both the gamete at faster rate. This technique have much advantage over the in vivo embryo production mode because it is being used in deformed cervix also. In deformed cervix, you can't use your precious donor through in vivo embryo production. You can't use teen tubal blockade condition, annular cow, cystic cow, prepubertal females, early pregnant animals, after calving, immediately after calving. So through what you call through in vitro embryo production, you can use this kind of problematic animals also you can take in your program, provided there should not be any intensive kind of the infection and that has reached to the ovary. If you see the global scenario of in vitro embryo production, it is switched over from in vivo to the in vitro from 2016 onwards. So even it's a trend now in vitro embryo production, and similar line government of India also pushing, and this time also in this budget outlay they have given 3,500 crores to strengthen the RGM, especially AI in vivo embryo production and other heifer rearing farms and so many so on. So this is our machine Dremensky, what we are using for the home pickup and we are WTA suction cum warming, and our wireless suction pump pedal pump is there. So we are using at our farm regularly I think every week. Very intensive programs are going on because of students research. Some are working on Tharparkar, some are working on Sahiwal, some are working on Buffalo. So, in this last from couple of months, I was a little bit busy for completing of their PG program in time. So, if you see how we are aspirating, so from live partially stimulated Sahiwal donor, we are aspirating the follicles, and that follicle will take to the lab, and we will grade these follicles in grade 1, grade A, grade B, grade C, and accordingly we'll put for the maturation over 24 hours. Thereafter, we'll evaluate the maturation process. So for in vitro embryo production, there are some process like we have to collect the oocyte from donor animal. Then you have to go for maturation. After that, you have to go for fertilization. Then you have to go for culture, and after culture of 7 days, 7 day or 6 day in case of buffalo, you have to evaluate your quality of embryo, and accordingly you have to transfer or freeze those embryo. So the program what we are following in case of cattle is 40 milligram for now it is 40 hour costing period. Going to already enter we are stopped stimulation of our indigenous cattle, especially Sahiwal and Tharparkar. We are not going for any stimulation. For buffalo, we are going for stimulation. And also, but in stimulation, what we found that in buffalo, though recovery bit improved, but quality drastically improved. So for buffalo embryo production, definitely you have to go for partial stimulation. But for our indigenous cattle, we need not to go for the stimulation or partial stimulation for pickup. So these are the quality of the oocytes. Grade A means 3-4 layer of cumulus will be there. Grade B only 2 to 3. Grade C some denudation will be there, and grade D and grade E. So A, B, C you put for the maturation. Grade D and E you can discard it in case of cattle. But in case of buffalo, you put all the oocytes, grade, whether it is grade A, B, C, or D. What we found that in the same pressure or even in reduced pressure, in case of buffalo, this cumulus cell attachment is very much fragile. Even during pipetting, you can see so many cumulus layers are reducing and reducing. So because of it's not because of quality, it may be handling and physical damage may be there. So it is always better to put, in case of buffalo, most of the oocyte for the maturation. You may get the embryo from that. So our Bos indicus breed tend to have more follicular waves and larger population of the small follicles. Bos indicus cattle have a larger number of the follicle per follicular wave and are more sensitive to the FSH. Because of higher sensitivity for Nellore cattle to the FSH, the total dose used to induce ovarian superstimulation is much smaller. Even you can go for 100 milligram is also sufficient as compared to our pure bred animal. So after that, you have to go for in vitro maturation. For in vitro maturation, you have to equilibrate the media some 2 hours before, and also you have to have a washing media and maturation media in 30 petri dish. Whatever coming the things from the pickup area, you have to wash in OPU media, or you can wash in Euroflush. After searching, you go for again washing, then washing in maturation media, then put into the washing media, put into the maturation media at your preset incubator. And what we found that benchtop incubator are much better than tabletop incubators. So if you are going for intensive in vitro embryo production, better to go for the benchtop incubator. Smaller incubator, tri-gas incubator are better than our earlier incubator, wherein you have to maintain 5 degree oxygen, 5 degree CO2, and 90% nitrogen at a 38.5 degrees centigrade. But in case of this one is for cattle. For buffalo, you have to have 38.8. So that will give the good maturation as well as more embryo production. So these after maturation, 22 to 24 hours, you will get means a flower, blooming flower-like structure all around of a cumulus cell layer around that this oocyte. So suppose if your donor is means far away, so now portable incubators are also available from WTA and other company. So you pick those 100 kilometers, 200 kilometers away, even up to 24 hour you put over there, means you pick up over there, and you put your oocytes for maturation into the this portable incubator, and you can have a journey up to the 20 to 22 hours. Then come into your lab, then take out a good mature oocyte, go for the fertilization, and then subsequently you go for the culture. So such kind of facility, flexibility are also available. Then you have to go for fertilization. Fertilization, then again, it depends on which kind of the media you are using. Whether you are using nitrogen media, serum-based media, or whether you are using cell culture-based media. Earlier, we are using IVF, so protocol is little bit different. Now we have switched over to the nitrogen media. So we are following their protocols, little bit modification here and there. And and for cement preparation is there. So cement preparation, again, you have to make a diluent, means diluted percol will be there, then sperm will be there. Then 200, you mix it and then pour over the conventional protocol, then cut open the two straw uh, on this one and go for the centrifugation at 1500 G for 6 minutes. And subsequently, you discard all the supernatant 100 microliters with suspension with particulate matter. You again mix it and wash with I pre-equilibrated IVF media. Again 200 G, 600 G for 2 minutes. Then discard the supernatant and you fertilize this your oocytes with 10 to 15 microliter of spermatozoa. So while you are fertilizing, you have to see whether sperms are moving quickly or not through microscope. If it is moving quickly, so you can expect a good fertilization rate. If it is sluggish, especially in buffalo, sometimes you will get very poor results. So these are the maturation. After maturation, after fertilization, you have to go for denudation to make presumptive zygote. Then presumptive zygote you keep into the after washing into the culture media. For culture media, you have to equilibrate for overnight or even 2 hours protocols are also 2 hour protocols are also there. And you keep into your incubator and don't disturb up to 6 day or in case of buffalo or seventh day in case of cattle and open it and potential blastos cyst, especially those with good quality of vacuoles inside, as well as zona thinning will be there, uniform inner cell mass will be there. So those are the very potential blastocyst. You go for the fresh transfer for calf conversion. So this is the culture more or less same. I have already informed you. You have to make the droplet either overnight or 2 hours before. Then whatever presumptive zygote, you denude it, wash it, then again wash into the culture media with same prefixed incubator for 6 or 7 days. You'll get the results, sure. So this the simplified way of oil must be there. No? Oil you have to keep according to your well size so that your none of the part of your droplet should be over the oil or should be otherwise there will be evaporation and loss of nutrient and your droplet may get dry also. So you don't expect any further embryo development. So one comparison we did whether stimulation is good or not. So what we found that stimulation, no doubt, it is a good because the genes related with oocyte growth and maturation are much expressed uh, as compared to the non-stimulated one. So and we started published this one also in zygote one and. Uh, study. So inspiration will get a uniform size of maturation as well as more blastocyst conversion will be there. That is sure. Uh, then again, the second and most critical factor is that recipient selection because ultimately whatever you are producing in laboratory, that doesn't mean that everybody can produce it provided with good media, good technician, better laboratory facility, quality CO2 incubator, you can produce the n number of the blastocysts. But that that doesn't justify completely your job until unless you are not able to produce the calf. For production of calf, selection of the recipient is much important. So you have to see that norm that should be regular cyclicity should be there, free from the infection, cervix should be smooth, And definitely on the day of the transfer, their CL will be good size, and blood flow must be more than 30%, and size must be around 15 to 16 mm. So choose those, and more so, choose from those lot wherein the recipient stress must be coincide with your IVF days. Though you can choose 12 or 24 hour earlier than IVF on the day of IVF, or 12 or 24 hour after IVF also. But definitely, when you are debating, you may get compromised pregnancy. But on the day of IVF is the most suitable day for the transfer. So, if you have Doppler ultrasonography, definitely you go for the blood. Yeah, and there is automatic software are there. Immediately they will tell what is the what is the volume or size of the corpus, how much blood flow. So blood flow should not be less than 40%. Mhm. Definitely, uh, is good. Sometimes your compromised transfer or little bit compromised recipient also could be managed. So each and every factor play a very important role in the calf conversion. And also this is the most crucial part, how you are transferring. If you are quickly transferring and transferring to the, if you see the transfer site, it should be cattle, especially our indigenous cattle or our crossbred animal, wherein you have to transfer at anterior one third of the uterus for better pregnancy. So for that, you have to have practice. It's not a theoretical job. So you have to go and practice vigorously and continuously. Then you could able to transfer properly and will get the get the respectable percentage of pregnancy. Otherwise, 1, 2, 3 you may get occasionally. So strategy to improve, there are a lot of strategy in book also. So wherein you have to increase the size of the PUF, you can rate growth of corpus luteum also, you can increase luteal phase progestin, you can increase the antiluteolytic stimulation by the conceptors also, you can increase, means you have to go for the antiluteolytic strategy. All those strategy from which you can increase the progesterone production, you can increase the donor synchrony, you can increase the nutrition of the donor. So all those aspects you have to take care very critically. So this one we have taken this follicular size-based synchronization protocol. So little bit, if animal having CL along with greater than 9 mm follicle, so you give a 24 hour before the donor PG. So that will come coincide with your IVF day. If it is a not IVF day, 72 hours in case of 24 hours before in case of MOIT program, and 72 hours before in case of IVF program. So like this, yeah, if it is a size is less, so you have to take more time. So sometimes 96 hours before you require to give before the IVF, or 48 hours in case of in vivo embryo production. So accordingly, you have to adjust. These are the freezing protocols. Our government of India also giving RGM this time also have funded this one. And some breakthrough research from India is that because now is getting all the media from the abroad only. So NDTV have some media that called Sasti, and it is effective. And also hormones also we are importing, and it is a comes under category of restricted drugs. So and this licensing and all is the problem with. So NDTV in collaboration with some other institute, they have developed a recombinant FSH, and that one is cheaper and effective also. But still, it's have to see the market. Uh, so that's problem. This one is otherwise by nature it is a 50-50% population. But because of lot of stray cattle, if you see that our northern India, the red is the more, and they are creating lot of traffic issue and so many crop issue farmers. And these are just So that is the problem. So that's why the sex semen technology come into the role. And the basic principle they are our X chromosome, X spermatozoa DNA is 4% heavier than the Y one. So that they are utilizing and they are segregating the X and Y spermatozoa. So this sex sorting, if you see the sex sorting station has been established and Uttarakhand, Gujarat, Madhya Pradesh, Uttar Pradesh, and Tamil Nadu. They have established and if you see sexing protocol, conventional methods are there, commercial methods are there, novel methods are there. At Ivory also people are working like microfluidic based sperm sexing technology, and they are working and also they are working with some drug where you can immobilize or reduce the motility of X spermatozoa so that you can separate the X and Y more efficiently. So these are the technology. And if you see that commercially flow cytometry are being used and wherein they are keeping 85 to 90% accuracy of semen sexing are there, and they are keeping only 2 million of spermatozoa. And it is a costly straw. If government subsidy is not there, it is costing around 1,200 to 1,500 per straw. And some limitations are there. Very costly equipments are there. Lower sorting speed and efficiency are there, and sometimes abnormal embryonic motility development is there, low pregnancy rate and fertility are there. So if you see in our India, only two companies are leading one. One is the genus breeding ABS, and second one is the ST genetics. So both are using the same principle, but the only difference is that ABS genus breeding India, they are killing the Y spermatozoa and keeping together. But ST genetics, they are lively separating both the spermatozoa in different manner. So through laser, they are sensing the here individual first they are staining the with hoist dye and they are passing through the narrow tubes and here they are detecting through sensor Y spermatozoa and through laser they are killing, but both comes in a single jacket. But in case of ST genetics, through charge, they are separate laser charge and all. They are separating the X and Y chromatozoa. Some spermatozoa are unsorted are there. So per call density, old method are there. Then modified SUMIP methods are there. Then identification of this antigen based also they are working. Volumetric differences are there. Then centrifugal counter current distributions are there. So now they are working on this particular resicumod. Resicumod agonist sex sorting. So what resicumod would do, they bind to the X spermatozoa without affecting the Y sperms and reduce the motility. And by uh, so the protocol requires specialized equipment, expert making applicable in laboratory condition fertilizer. So this one is the good. If it is successful, definitely is very much helpful. And then the same things which I have already explained. And third one is the third or fourth one you can say it's animal cloning or somatic cell nuclear transfer, wherein we are identical copy, genetically identical copy of the individuals. So, first Dolly sheep was the born on July 5th, 1996. Dolly was the first mammal to be cloned from an adult cell. And British scientist Ian Wilmot and Keith Campbell in 1996, they have produced Dolly using somatic cell nuclear transfer. What is somatic cell nuclear transfer? Donor nucleus is picked inside the enucleated oocyte and electrofusion is there. Then reconstituted zygote formed, equivalent to be a fertilized ovum with potential to develop into the blastocyst if implanted develops into offspring. Hence, clone is identical to twin of the donor animal. So there are two types, micro manipulator using and handmade guided cloning. So our NDRI and CRB, they are using handmade guided cloning. Otherwise, here steps are there. Like you have to collect the oocyte from the donor animals, and at the same time you have to collect the somatic cell from the donor animals. So then we are maturing the oocytes. I think here, yeah. So you have to collect the oocyte, preferably from slaughterhouse, then mature it. Then after maturation, you have to go for trypsinization. You have to remove the zona and all. After removing the zona, you have to cut open the polar body, this nuclear portion. So you have to take only cytoplasm. You have to take only cytoplasm, and then at the same time you are processing the somatic cell also. You have established the culture, single layer culture, single cell suspension. Then take the fuse cell. With the help of electrode, you fuse this oocyte with the somatic cell. So nuclear material from the somatic cell, you fuse with electrodes, and there will be gamete formation, and then gamete formation will be there. Then you go for culture. So fusion, then culture will be there. Yeah, this one. One cell, two cell, eight cell, 32 cell, and then compact morula. And when it become a blastocyst, sometimes seventh or eighth day, depending on your timing of IVF. So this one, so in this our NDRI have very much leading. They first produced the buffalo clone in Samrupa in February 2009 using the donor cell ear fibroblast, and died in the 7th day of birth. Then they produced a Garima 2 in 2010. Then they produced Rajat, and then they produced the Dipasa. Means their donor cell used to be different. Sometimes ear fibroblast, sometimes memory, sometimes dual cells they are using. And some Mahima they produce, they born to clone. Means from Garima, clone Garima, they have produced Mahima also. First clone indigenous Giri female produced at NDRI. So, and the same second leading institute is the CRB. They have produced a 9 very much elite Murrah male buffalo, and they are all in the semen collection. They are all in the semen collection, and very prized bull. So NDRA and CRB are the leading, and these are their history. And these some of the problem, definitely cloned animals. Some pregnancy rate is quite low, abortion rate is more, fetal and placental abnormality is to be there, large offspring syndrome chances are there. Yes, sir. Changes will be there because cytoplasm, the oocyte contain lot of things. So and these are there. Mm. Then, if you see, talk about the transgenic technology and method, principles, and application. In that context, Government of India also given a very big project network program on application of a genome editing technology for improvement in livestock health and production. So there are, I think, some 80 crores they have given, 12 collaborating centers. Some are working on health aspects like vaccine production through editing and all, and some are working on the production aspects, especially NDRI, CIRG, CIRB, they are working production aspects. So transgenic animal are produced by the transfer of the foreign gene or DNA into the genome of other animals. The DNA gene that is introduced is called transgene, and the overall process known as transgenic technology or transgenesis. So these are the methods how you are introducing the particular gene of your interest into the genome sequence of animal. Mm, mm, so through now they are using CRISPR-Cas in here our lab also they are using CRISPR-Cas. Inserted gene may have means some disadvantages are also there definitely, may have multiple function. Many embryos animal die due to the change in the direction of evolution. It may lead to the mutagenesis and functional disorder. Mosaicism will be there. Low survival rate will be there. Transgenic efficiency is very poor. Technique is very costly and require high skill manpower. So in 2020, this Emmanuel Carpentiers and Jennifer Doudna, they have been given Nobel Prize in 20 in chemistry for discovery of this CRISPR-Cas9. Definitely in gene editing, it is a breakthrough research, and it's very easy to do with this CRISPR-Cas micro injection. And this one, it's a very easy to set up a molecular laboratory. Reagent are not so expensive, and quick multiple single guide RNA offer multiplex editing and gene microinjection of the Cas9 protein inserted into the mRNA has proven to be the more efficient to obtain HDR in the mice rat. This one mosaicism in the founder edited animals produced from the injected zygote. So sometimes we may get result, sometimes we not get also. Because we tried this CRISPR-Cas9s and we have transferred 75 goat embryos. But so far we didn't get any pregnancy. And then generation of beta lactoglobulin knockout goat using CRISPR-Cas9. The multiplex gene editing CRISPR-Cas9 exhibit desirable muscles hypertrophy without detectable off-target effect in sheep. So this aspect muscular hypertrophy, they are working both CARG and CAR NDRI, they are working. So our conclusion is that ART has potential for faster multiplication of superior germplasm, but it is a costly and required highly trained person. Thank you. Thank you all participants and host. I think I was finished in time. I a bit late.
Thank you, sir. Thank you so much for presentations. And it was indeed a very wonderful and informative presentation. And participants have liked your presentation so much. And we have got the response from so many participants.
I will share this presentation to Dr. Dinesh, ma'am.
Yes, sir. You can share the
So
presentation.
interested can have
Yes.
from this coordinator.
Yes, sir. Thank you, sir. And also we have one or two questions if you would like to answer.
What question, ma'am? I'm not able to see where it is
Yeah,
in chat
question
box.
is how easy or tough it is to do it at a field level, and what are the challenges the field veterinarians are facing.
Ma'am, you are not audible. Either you speak little loud or you keep your microphone bit near.
Sir, how easy or tough is to do it in the field conditions, and what are the challenges?
For what? For this embryo transfer.
Embryo transfer.
No, ma'am, many states, especially if you go to the Gujarat, Uttarakhand, and so many private partners are also coming in this field. And so embryo transfer, IVF embryo transfer, they are doing, especially in a light animal where organized farm are more. The problem is that still our freezing protocol not has been properly standardized. Once the freezing protocol is standardized, definitely the success will be much more and it will compete with AI.
Okay.
Definitely, transfer is not going to take its own space over a time, and slowly it is progressing.
Yes, sir. And so what is the success rate of IVF and OPU techniques?
Ma'am, what at our farm, though we have conducted at a small scale, it's around 45% pregnancy we are getting in farm condition, and field a little bit 10% dip is there, 30% we are getting. Because in field condition, we don't know actually what is the recipient estrus time with our donor in respect to the donor. So that one of the factor, and more so sometimes we have to travel 1 hour, 1.5 hour, so embryo will get compromised. So what is the biggest challenge at present is that freezing should be standardized and it should be transferred in canister, and so that you thaw it and transfer it. Again, for thawing, we have to have a microscope, and that to stereo zoom microscope.
Okay.
But it should be practiced in where dairy business is more intensive, where farmers prefer to have high yielding animals. Even if you give the high yielding animals to the farmers of Jharkhand, Eastern Uttar Pradesh, Bihar, they can't able to maintain. If you give the high yielding animals of Punjab, Haryana, where their baseline starts from 30 plus. Here, very rarely in this our states where resource poor farmers are there. So that we look. That's why in these states, especially now, from large animal practice to the large animal rearing to the shifting to the small animal rearing, and we are also promoting same things. through our various project and program, because feed fodder is also becoming too much costly. This bhusa become 12 rupee and our concentrate becomes some 39, 40. So that's all. And the milk prices are more or less same, 30, 40. In villages is still 30, 35. So it's not costly business, but okay, people have no options, so they are practicing.
Okay, sir. And sir, what are the selection strategies to raise a recipient for embryo transfer based on presence of CL on ovary?
Uh, uh, See, for beginning, you have to have depends on history. Where it should be the regular cyclic, should be the free from the infection. While palpating, you should palpate the cervix also. Cervix should be smooth, and organ should not be too much pendulous. One thing, and in very beginning, and at the day of transfer, suppose you have synchronized 10 to 15 recipient, and you are going to transfer in 12 animals. 12 animals you have finalized. Out of 12, you are going to transfer 8 or 10 or 6. So in at the day of transfer, you have to see the size of corpus luteum, blood flow to the corpus luteum are the two important parameter. If size is somewhere 14 to 16 and blood flow is more than 40%, so that is the ideal recipient. Provided that should not have any obstruction in the cervix and organ should not be too pendulous.
Yes, sir. Thank you, sir. And with this, we are done with the questions. And thank you for the informative
Okay.
session.
No, ma'am, thank you to me. Thank to all your organizing team, especially Dr. Dinesh. Very short notice he had told sir कुछ लेक्चर ले लीजिए तो मैं कहा कोई बात नहीं व्हाट आई एम डूइंग आई विल डिलीवर ओवर
Yes, so you have explained it very well. It was very informative session for all of us.
yeah, ma'am. Actually, our so many PG and PhD students are very much interested for this technique. Though here less scope for research, it is already well thorough and standardized tech tools and techniques. But they are forcing me to sir continue this area. Otherwise, these technology will die from India. So one aspect is that one, and second, those center who are offering the training for these aspect, they are charging very high. For 10 days training, none of the center they are costing less than 1.5 lakhs. So from where a normal veterinarians are those who are not in these jobs, so how they are going to get it? So for that,
Okay.
every years we are training 10 to 15 our students as well as vets also with a minimal cost.
Thank
Okay.
you,
Thank you,
sir.
sir.
Thank you for giving your precious time.
Thank
Okay.
you, sir.
Thank you, whole your team, especially organizing that this kind of international seminar. That to the fag in the financial year, a lot of other pressures are there to settle all those bills and all. And more so for
Definitely.
Indian point of view, this timing is also na evening and late evening is time for other works. So no problem. Things will move like this only, and we should progress like this only. So once
Thank
again, from bottom of heart, thank you to all organizing team, especially Dr. Dinesh, Dr. Imani, Dr. डॉक्टर साहब डॉक्टर आई थिंक सामंत अभी
Jitendra
कॉल
ji.
की क्या हाँ कॉल किए थे सो होल टीम and look forward if in future also if any opportunity either in practical way or in theoretical aspects I will happy to be with you.
Thank you, sir.
Definitely.
Thank you, sir.
Thank you once again. Okay, I
Thank
will
you.
share the PPT Dr. Dinesh to you.
Okay, sir. Okay, sir.
Okay.
Okay, sir.
So everyone, we have reached to the end of today's session. So on the behalf of Vital Biotech Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Science and Animal Husbandry, Ranchi, Birsa Agriculture University, Ranchi, and the entire organizing team, I deliver my heartfelt gratitude to our amazing speaker, Dr. Abhishek Gupta and Dr. Brijesh Kumar, for their insightful talk. I'm very sure that their hard work, experience, and presentation, their knowledge must have motivated and for joining this 7 days training program and being so patient and attentive for today's session. And participants, as you that tomorrow is the last day of this training program, so I request all of you to must join tomorrow's session as we have to make some important announcements tomorrow. So we'll see you all tomorrow at 6 p.m. Thank you all and have a great evening.
Thank you all.
Thank you.